Good evening, how are we all? I hope we're all fine and well and dandy. Welcome to our virtual clinic this evening on Doyne's Honeycomb Dystrophy with Jacqui Van Der Spuy from the UCL in London. It's a very sort of very small selected group this evening because it's quite a rare condition, which is good for us because I'm looking forward to learning lots about it. Just before we get on with all that though, there's a bit of housekeeping. So just to let you know who I am, if you don't know, my name is Colin Daniels and I'm the Working Age and Young People Service Manager for the Macular Society. I've been in post for a couple of years and we've set up this lovely service that's aimed at people who are just a little bit younger than the usual people with macular disease. So sort of people that are still maybe still be working or parents with kids with macular dystrophies and young people. The little bit of housekeeping I talked about, if you can just make sure that you leave yourselves on mute, he says, stumbling over his own teeth, and your cameras off, and please try not to share your screen. It does happen from time to time, so just sort of be aware, be careful not to do that if you can. Not to worry, my support worker Patience is around, so if there's if she's sort of keeping an eye on things, you accidentally share your screen, she'll sort of turn things off, so don't panic too much. Also on the call, we have Jerry Hode, who's the research manager from the Macular Society. And at the end, she'll just say a few bits about what's going on with that sort of things. We just want to say hello, Jerry.
Yeah, hi everybody. Yeah, I'm the research manager at the Macular Society, and Jacqui's project that she's going to be talking about is one of our research projects that we started funding this year. Thank you.
Brilliant, thanks ever so much. And so I mean, you're not really come to listen to me, really. You've come to listen to Jacqui. So I introduced Jacqui. So this is Jacqui Van Der Spuy, sorry, from UCL. I'm sure she'll introduce herself a lot better than I do, and she's going to talk about her research and how it can support not just potentially Doyne Honeycomb Dystrophy, but other dystrophies as well, macular dystrophies as well. So good evening, Jacqui.
Good evening, Colin. Thank you so very much. Thank you to you, yourself and Jerry. It's really a great honor to be here this evening, and I'm really delighted to be able to talk to you all about this project that has been sponsored, which I'm very grateful to the Macular Society for sponsoring. So I have actually prepared some slides today, however, they're not essential for you to be able to follow the talk. It's just really a prompt for me. And I think that Colin has let me know that it's absolutely fine if anybody would like to ask me questions during the talk. I'm perfectly happy to answer questions during the talk as well. So please don't feel shy to interrupt me. I'm perfectly happy to speak with you. So I'm going to try to share my screen now. Let's have a look. Okay, so today obviously I'll be talking about a research project funded by the Macular Society, which involves investigating therapeutic prospects for a disease called Doyne Honeycomb Macular Dystrophy, or Doyne Honeycomb Retinal Dystrophy, which I'm sure you all know a lot about already. But as you know, this disease forms part of a group of diseases that are juvenile macular dystrophies. And essentially what characterizes them is that they have the onset of symptoms in early to mid-adulthood. So in the 4th or 5th decade of life, patients have the onset of the symptoms, although it can happen quite a bit earlier as well. And the hallmark of this group of diseases is the accumulation of deposits of protein and lipid underneath the layer of cells in the eye called the retinal pigment epithelium. So to begin with, I just want to remind us a little bit about the structure of the eye and some of the clinical phenotypes or the phenotypical clinical symptoms of the disease as well. And so we know that the eye consists of a number of different layers. The innermost layer of the eye is the neurosensory retina, and just beneath the neurosensory retina we have a layer of cells called the retinal pigment epithelium, and beneath that is the choroid, which basically houses the choroidal vasculature, and then beneath that there is the sclera. Now, the retina itself is the bit that is responsible for our vision. So it's the bit that's responsible for converting light into an electrical impulse, which is then conducted via the optic nerve to the brain. And within the retina, there are a group of very, very special cells called photoreceptor cells. And there are two types in the human retina, rod photoreceptors and cone photoreceptors. And the rod photoreceptors are responsible for our dim light vision, and cone photoreceptors for our vision in bright light. So they're responsible for our color vision, or our visual acuity, if you like. And these are very special neurons because they have a special extension, or a cilium, which consists of something called an inner segment and an outer segment. And all the machinery that is required to convert light into an electrical impulse is housed within this special outer segment of the photoreceptor cells. And within the retina, there is also a very special area of the retina called the fovea. And within this area of the retina, at the central bit of the fovea is a bit called the foveal pit. And in this area, there are no cone or rod photoreceptors at all. So this basically is the blind spot in the retina. And then within the fovea, we have basically the cone photoreceptors are arranged at their highest density within the fovea at the exclusion of rods. So in the fovea, we have a huge high packing density of cone photoreceptors without any rods. So that's the bit of the retina which is really responsible for our color vision, or for our visual acuity. And then outside of the fovea, the density of cones rapidly drops off, and we have more rods than cones. And these bits of the fovea, so the foveal pit and the foveal slope and rim and the foveal rim, all together cause something called the macula. And it is this bit of the retina that is primarily affected in these juvenile macular dystrophies. However, the primary defect itself isn't in the retina, but in this layer of cells called the retinal pigment epithelium. So the retinal pigment epithelium is a very special layer of cells just beneath the photoreceptors. These cells arrange themselves in a monolayer, and they are tightly packed together or joined together by something called tight junctions. And in this way, they form a very, very tight barrier. They essentially are responsible for maintaining the integrity of the blood retinal barrier, if you like. So on the one side, they have photoreceptor cells, and on the other side, they have the choroidal vasculature. So, you know, they're really important for maintaining the homeostasis, if you like, of the photoreceptor cells. They bring nutrients to the photoreceptor cells, and they clear waste away from the photoreceptor cells. And in addition to this, they are highly polarized cells. So on one side, they have at the top, they have what we call an apical membrane. And extending from this apical membrane are a number of microvilli which interdigitate between the outer segments of the photoreceptors. And actually, early in the morning and late at night, we actually shed a large portion of our photoreceptor outer segments. And so these RPE cells are responsible for engulfing these outer segments, if you like, or phagocytosing them and then clearing them away. And the RPE also participate in the visual cycle itself. So they're really important in terms of maintaining the homeostasis of the photoreceptors. And in Doyne Honeycomb retinal dystrophy, atrophy of this RPE layer occurs. And this atrophy of the RPE layer then affects the homeostasis of the overlying photoreceptors, which then results in the clinical symptoms that patients experience. And so over here, essentially this, I've shown here a fundus photo, and it's basically just a photograph of the retina itself. And it essentially shows the accumulation of these drusen underneath the retinal pigment epithelial layer. And in patients, they're normally radially distributed in the macular area and around the optic nerve. And they start off as quite small deposits, which then expand with age and eventually join up to form a honeycomb pattern, hence the name of the disease, Doyne Honeycomb retinal dystrophy. And I've also shown a picture of something from a technique called optical coherence tomography, which is essentially a cross-section through the fovea. But all that it shows is this mass accumulation of these deposits underneath the RPE, which then also results in atrophy of the overlying outer retina, and hence the clinical symptoms. So what are the clinical symptoms? Patients obviously experience a loss of central vision, so loss of central visual acuity. And also quite often patients experience or develop scotomas, which are kind of gaps in the vision. It's a breakdown of vision in the visual field. And patients also experience dyschromatopsia, which essentially is where colours begin to look dull, as well as metamorphosia, which is where straight lines start to look wiggly or curvy. So in the case of Doyne Honeycomb retinal dystrophy, this disease is caused by a gene called EFEMP1, and this particular gene codes for a protein called fibrillin 3. And so fibrillin 3 is actually one of several fibrillins that we find in humans, or a group or a family of fibrillins. And the fibrillins are really interesting. The proteins are characterized at one side by something called a fibrillin type domain. They are also characterized by a tandem arrangement of consecutive domains, which are called EGF, or epidermal growth factor domains. And in fibrillin 3, there are 6 of these domains, and the mutation that causes the disease is found in the 6th domain. At the other end of the proteins, there is something called a signal peptide, which is responsible for the secretion of these proteins. So these proteins are really interesting. They're normally expressed in cells, but then they are secreted out of cells into something called the extracellular matrix. And the extracellular matrix is basically the glue, if you like, that holds tissues and cells together. So these fibrillins are really important for tissue integrity. And because of that, we find that mutations in family members of the fibrillins are often associated with defects, for example, in connective tissue. For example, mutations in the gene for fibrillin 1 cause a disease called synpolydactyly. And mutations in the genes for fibrillins 4 and 5 cause something called cutis laxa. Cutis laxa is where there's a loss of elasticity in the skin, so it kind of just hangs down in folds. And then there are genetic variants in the genes for fibrillin 5 and fibrillin 6, and these actually increase the risk for the development of age-related macular degeneration. And then, of course, we have fibrillin 3, and mutations in the EFEMP1 gene cause Doyne Honeycomb retinal dystrophy. And so normally in cells, the normal or the wild type copy of the gene is, or the protein is expressed in cells, and it's processed through an organelle in cells called the endoplasmic reticulum. And normally it's processed through the endoplasmic reticulum and then secreted out into the extracellular matrix. But what happens with the mutant protein is that it misfolds, so it can't really acquire its normal shape or conformation. And then it accumulates in this organelle called the endoplasmic reticulum. And then what happens is that it actually induces a cascade, a stress cascade, if you like, which is called the unfolded protein response. And this in turn results in the deposition of these kind of lipids and proteins just underneath the basement membrane of the retinal pigment epithelium. So what's interesting about Doyne Honeycomb macular dystrophy or retinal dystrophy is that it is inherited in an autosomal dominant fashion. So just to build up a little bit on that, I'll just go on to describe autosomal recessive disease in the first place. So in our DNA, in our genome, in our nuclei, we obviously have our DNA, and within our DNA, we have 2 copies of every single gene. So we always inherit 1 copy of a gene from our mother and 1 copy of the gene from our father. And what happens in recessive disease is that for an individual to become affected, they inherit a mutant allele from, or a mutant copy of the gene from both the mother and from the father. So both of their copies of their gene are defective. And generally what this does is it results in a complete loss of function of the encoded protein. So these are often called loss of function diseases. However, Doyne Honeycomb retinal dystrophy is an autosomal dominant disease. And in the case of autosomal dominant diseases, essentially, for a patient to be affected, they need inherit only a single copy of the faulty gene from either the mother or the father. So in patients with dominantly inherited disease, they actually have one normal copy of the gene and one faulty copy of the gene. And it is thought that the faulty copy of the gene actually results in a gain of function toxic effect. So what it does is it imparts on the protein that it encodes a toxic function that it normally wouldn't do in the cell. So yes, so Doyne Honeycomb retinal dystrophy is inherited in an autosomal dominant fashion. So patients receive one faulty copy from either the mother or the father. And this has helped us to kind of inform and think about the kind of therapeutic strategy that we want for Doyne Honeycomb retinal dystrophy. And that is basically the premise of this project, where we want to actually use a therapeutic strategy to target the faulty copy of the gene and to downregulate the faulty copy of the gene and thereby remove the faulty protein, if you like. And there are a number of things which have recently made this kind of project accessible or possible. And the first of these is that there have been some really significant advances in the field of stem cell biology in the first instance, for example. And I'm sure that many of you will know or will have heard that what we are able to do nowadays is we are able to take cells from an adult, skin cells or cells from the urine or blood cells, and we are able, in this case I've depicted here that we will take a biopsy, so skin cells from a patient, and then what we can do is we can treat these skin cells with something called Yamanaka factors. So essentially these are just special transcription factors, and what happens when you treat these cells with these transcription factors is that you can reprogram these cells into something called induced pluripotent stem cells. Now, induced pluripotent stem cells are really special. As the name implies, they are pluripotent, which means that they have the ability or the capacity to form any cell or tissue type in the adult human body. And so what we're able to do is we're able to take these induced pluripotent stem cells and we can grow them under very special conditions so that we can turn them into a monolayer of retinal pigment epithelial cells. So an RPE layer in a dish, if you like. And this technology is really powerful because what this means is that we can produce RPE cells from a patient which are genetically identical to the patient, him or herself. So if the patient harbors a mutation, for example, then the iPSC RPE that we derive from that patient will similarly carry that mutation and will manifest the disease phenotype. And this is really important because obviously it's not possible to take the retinal pigment epithelial cells directly from a patient's eye. And so these iPSC RPE cells are a very powerful platform for testing new therapies and also for exploring disease mechanisms. We can obviously also look at RPE cells in post-mortem tissues. However, then we're looking at the end stage of disease and we can't really understand how we've gone from a normal to a pathological situation. So the iPSC RPE are really a powerful platform for understanding disease and developing therapies. And this, I've just shown a slide here, which basically shows that these iPSC RPE are akin to the RPE that you would find in an adult human eye. So if you look at these RPE cells from above, they form a tightly packed hexagonal arrangement of cells which are highly pigmented. And if you slice through these RPE in cross-section, they have an apical, you can see that they are polarized, so they have an apical membrane and a basolateral membrane, and they have these accumulation of pigments on the apical side and microvilli. And we can also label these cells with markers for the apical and basolateral membranes to show that they are highly polarized. So for example, we can label them for a marker called ZO1, which is at the apical membrane, or a marker called BEST1, which is at the basolateral membrane. And we can see a mass accumulation of melanosomes loaded with pigment on the apical side as well. So it's really important that these iPSC RPE in a dish are exactly as RPE cells would be in a patient. And the other thing that I mentioned before is if these RPE cells are derived from a patient with a mutation, then the iPSC RPE in a dish can manifest the disease phenotype. So of course, I have shown a slide here. Essentially what it shows is that iPSC RPE from the patient look normal morphologically. So from above, they have this hexagonal packed arrangement together, and they are also polarized. So they have an apical membrane and a basolateral membrane. But the big difference is that the RPE derived from patients with a mutation in EMP1 accumulate these big blobs underneath the RPE layer. And these blobs consist of proteins and lipids that we would normally find in drusen in RPE patients. So essentially, these iPSC RPE from Doyne Honeycomb retinal dystrophy patients can actually manifest the disease phenotype. And so the goal of this particular project is to develop a therapy to see whether we can then rescue this disease phenotype, i.e. treat the iPSC RPE with the therapy and see if we can resolve the formation of these drusen in the model. And so the therapy that we will be using in this project is a therapy called an RNA-directed therapy. And I just wanted to quickly remind you what RNA is. And so in our nucleus, as we know, our genes are contained within the DNA, and the DNA encodes all the information to make the proteins in our cells. However, it goes via rather circuitous routes. So we have our DNA, and our DNA is first, or our genes in our DNA are first transcribed to something called RNA, or pre-messenger RNA, which undergoes a little bit of further processing to form something called messenger RNA. And the messenger RNA itself then is used to synthesize the protein. So in the RNA-directed therapy, the molecule that we're targeting is actually the mRNA transcript. And in this kind of therapy, what we're actually doing is we're actually hijacking a system in the cell that normally comes into play when our cells encounter foreign genetic material, for example, from a bacteria or a virus. And what this approach involves is that we synthesize, or we make a synthetic piece of DNA called an antisense oligonucleotide. And this antisense oligonucleotide is complementary to the mRNA transcript that we wish to target. So essentially, we synthesize the DNA oligonucleotide, we deliver it into the cells. The DNA oligonucleotide will then bind to the RNA transcript that we wish to target to form something called a DNA-RNA hybrid. And this DNA-RNA hybrid is essentially recognized as foreign by the cells, and an enzyme called RNase H comes along and recognizes the DNA-RNA hybrid and chops up the RNA transcript so that there is no protein produced from that transcript. So the theory of this in our particular project is that we are going to design an antisense oligonucleotide which specifically targets the mutant allele. So the antisense oligonucleotide itself harbors the mutation so that it is complementary to the RNA transcript that harbors the mutation. So here we can have our DNA antisense oligonucleotide, which can bind directly to the RNA transcript. It's recognized by RNase H, and then we preferentially have degradation of the mutant allele so that the mutant protein is not produced in the cells. So this is kind of approach is called allele-specific antisense oligonucleotide targeting. And so what we will do is we will apply these antisense oligonucleotides to our iPSC RPE model and evaluate how well they will be at downregulating the mutant allele and then resolving the phenotype, which consists of the drusen. So that's the primary strategy that we're going to employ in this particular project. However, just to say that there is another strategy, I'm not sure how many minutes I've got left, and I'm not sure if I've got time to go into it, but there is another strategy that we wish to employ in this project, and that is where we can use antisense oligonucleotides to target both the normal allele as well as the mutant allele. So we could knock down both the copies of the gene. And the reason why this might be successful is that it has been shown in animal models and also in patients that if you have the loss of the EMP1 gene in the RPE, that this is actually protective against the formation of drusen. And in fact, these RPE do not manifest a phenotype if you lose the gene completely. So there's actually two different strategies that we can employ in this particular project to try to rescue the disease phenotype. So I guess the big question is, what's the translational potential of these ASO to patient benefit? I think that the translational potential is quite high. So essentially there are a number of ASO therapies which already have been approved by the FDA and the EMA, and that are used to treat patients with different diseases, including Duchenne muscular dystrophy, spinal muscular atrophy, and familial amyloid polyneuropathy. So these are different diseases where antisense oligonucleotides have been approved for use. And indeed, I think there are a large range of antisense oligonucleotides that have entered early phase clinical trials, or even late phase clinical trials. One of them is called QR1110, and QR1110 is an antisense oligonucleotide designed to treat a disease called Leber's congenital amaurosis, so a retinal disease. So there is a large precedent for the successful use of ASOs to treat disease, and so we're hoping that with the use of our human model that we can, first of all, provide proof of principle that this ASO therapy can work for Doyne Honeycomb retinal dystrophy. And the interesting thing is that a lot of the other juvenile macular dystrophies are similarly inherited in an autosomal dominant fashion. So we hope that if we can prove that this works in this particular type of juvenile macular dystrophy, that hopefully it's a technology that we could apply to other forms of autosomal dominantly inherited macular dystrophies as well. And then I just wanted to say that this project is basically taking place it's a team effort, essentially. We have obviously this fantastic support from the Macular Society, but we've really accumulated a really excellent team of experts to help with this project. So in addition to myself, there's Dr. Amanda Carr, Professor Mike Cheetham, and Professor Pete Coffey, all at the Institute of Ophthalmology, which are helping us to achieve the goals of this particular project. So I think that I have flown through all of that really, really quickly.
No, you've done about 25 minutes. I have been keeping an eye. So.
Oh, good. Okay.
Yeah, that's great. Yeah.
So I'm really, really happy to answer any questions that anyone might have for me. Shall I just stop sharing my screen?
Yeah, stop sharing your screen and let's see if we've got any questions. So because there's not many of us this evening, I think we'll just let people unmute themselves and ask a question. Just be aware that when you ask a question, because your camera's not on, your name will come up. But patients, when we edit this to put it on, to post it later, patients, my support worker will blank out all your names, so they won't come up as part of the recording. Just be aware of that. So has anybody got any questions for Jacqui? Definitely silent.
Don't be shy.
Yeah, don't be shy.
Shall I've got a question, Jacqui. It's Geraldine here. Do you know? Have you got any idea how many people actually have Doyne Honeycomb dystrophy? Do we know the rate? I know these things are really difficult because they're so rare.
Yeah, it's a really difficult one. It is really rare. I think that the prevalence is 1 in 30 to 80,000. So in terms of absolute numbers, I'm not 100% sure, to be honest with you. But I think that where we are lucky is that we have a large number of patients actually at Moorfields Eye Hospital. So I feel quite bad now because actually my other collaborator at Moorfields Eye Hospital is Mike Michaelides, Professor Mike Michaelides, and he has actually done quite a large clinical study on the on patients with this particular mutation at Moorfields Eye Hospital. So this is the patient cohort that we will be obtaining skin samples from for this particular project. But yes, it is rare. So as hard as I've tried to find out, mostly everywhere it says numbers unknown.
It's always are you still looking for cohorts for any research that you're doing?
Yeah, I mean, we are always interested in cohorts for sure. And we do, as I said, we do have a large cohort of patients at Moorfields Eye Hospital, about 30 or more patients or so. But the other aspect of the project that is quite interesting is obviously that there is some variability in the severity of the disease amongst patients. And so there are a number of reasons for that. There might be epigenetic factors, so other genetic factors which determine the severity of the disease, and also environmental factors, which I think is similar to and overlaps with things like age-related macular degeneration. So things like smoking, etc., are also thought to have an impact on the disease. But an interesting question for us is what those other extragenic factors might be. So, for example, for this particular project, we will obviously recruit patients with the severe disease phenotype so that we can manifest that disease phenotype in our model, if you like. But also, it would be interesting to look at a range of phenotypes across patients to try to see if we could understand more about what the causes are, maybe the epigenetic factors that are responsible for that phenotypic variability, so that difference in the disease severity amongst patients. So yes, so we are always interested in looking for and recruiting new patients for our studies.
Brilliant. So I'll put it out there again. Anybody got any questions for Jacqui?
Yes, I have a question.
Great.
It's Amy here. So I just wanted to know at what sort of stage would this type of therapy sort of be administered? Because obviously it's an inherited disease. So you know perhaps before someone showed symptoms that they might get it. So would it be when you sort of know sight loss is occurring or much earlier on?
Yeah, that is such a good question. And I think this is something that would definitely require a large amount of workup. As you say, it's autosomal dominant. So if it is inherited in a family, obviously there will be, siblings might opt in to find out whether they harbor the mutation and therefore might develop the disease. So there's a question of whether it's treatment for prevention or treatment for cure. And of course, it's difficult to know how effectively these ASO will work after the onset. So you have to be careful because I guess you reach a point where the RPE atrophy is too severe. So in my mind, I think treating earlier, prevention rather than cure, or a combination would probably be a better option. So we do know that the drusen do form early on in these patients as well. But most of these therapies, the important thing is that you treat whilst the morphological whilst the tissue itself is still morphologically preserved. So, you don't really want to treat once the atrophy of the RPE or once the effects on the overlying retina have begun. So probably earlier would be better, but we do have ways in which we can test that. So obviously there are, for example, a number of mouse models. There is a mouse model of dominant disease caused by EFEMP1, which recapitulates the human disease. So we could, treat mice early on or after or later on, and to evaluate the effect of ASO in early or later disease, for example. But the only thing is that the mice model is a tricky model because mice are mice in the first instance. So mice, for example, do not have a macula, they're nocturnal. So although they have the deposition of drusen, they don't actually have some of the features that the human retina does, for example. But I think that is really an interesting question. And, in the process of translation, that's definitely something that would have to be teased out a lot better. So do we treat before the onset of drusen or just after the onset or well before atrophy of the tissue itself? It's a really great question. Thank you.
Just as a follow-on, would it be a one-off treatment or would it have to be repeated?
Yeah, so at the moment, most of these treatments are used as one-off treatments, interestingly enough. But that is, again, so, then a lot of things come into play, like the pharmacokinetics and the pharmacodynamics. So how long does the ASO hang around for? How long does it take before it's cleared? All of those from the body, all of those sorts of questions then come into play. How long does the effect last for? So at this point, it would probably be quite difficult to answer. So, if we wanted to go to translate to patients, then we would have to do a very detailed pharmacokinetic and pharmacodynamic study to determine that as well. Most studies would do this. They would give a one-off treatment, and they would also have a treatment regime where you would treat a number of times as well. So yes, also a great question.
We've had one come in through the live chat, which is what's the best way to diagnose and what are the best scans to use?
Okay, so the best way to diagnose, I would say instantly that as it is an autosomal dominant, it is most likely that, you know, if your mother or father have it, then it's likely that the offspring may have it as well. So genetic testing, I think, is important. So if parents have the disease, then the children could opt in for sequencing, so simply taking a DNA sample and sequencing to see if they harbor the EMP1 mutation. So that's in terms of genetic diagnosis. And in any case, it's always really, really important to confirm the genetic cause, because that helps you to then identify the kind of juvenile macular dystrophy that it is. So clinically, some of these dystrophies appear quite similar, but it's only when you do the genetic testing that you understand actually what subtype of the disease you actually have. So I would say that the first port of call would be to do the molecular genetic diagnostics to confirm, you know, what the disease, which of the genes is causing the disease, and therefore the subtype of the disease that the patient might have. I think there was another bit to that question, wasn't there?
Yeah, it was which are the best scans to use?
The best scans. So yeah. I showed you a couple of photographs there, and there is a very typical characteristic. So the fundus photo is just a photograph that you take of the retina. You shine light on the retina essentially, and you take a photograph. And the formation of drusen in Doyne honeycomb retinal dystrophy is quite typical. So you have this radial pattern of drusen, and they come together in this honeycomb kind of pattern. So that could be very diagnostic for Doyne honeycomb retinal dystrophy. So that's the fundus photograph, but also the OCT that I mentioned before, this optical coherence tomography, is also take an image that is taken in live patients. Essentially, it's a technology where you shine light through the retina, and obviously the light is reflected back, but depending on the density of the tissue that it strikes, so you can actually see in cross-section the different layers of the retina. And this is informative because it can show you the accumulation of drusen under the RPE, but it can also show you whether there's been any damage, you know, to the RPE itself or to the overlying retina. So I would say a combination of those factors, of those kinds of scans, would be useful and diagnostic for the condition.
I do like an OCT. I do. I mean, I can't see them very clearly myself, but they're just so impressive. It really ticks my geeky bubble button, really.
Yeah, they are incredible. And obviously this, I think, you know, the things that are happening in terms of the advancement of imaging is absolutely incredible at the moment. So, you know, AI for diagnostic imaging, for example, is coming on and will come a long way to help with these sorts of diagnoses. But yeah, I would say the fundus photo and OCT.
Yeah, brilliant. So has anybody got any other questions? Be brave, go on. I know you want to. You don't have to. It's not important. Well, it is nice. Okay. Well, Jacqui, if no one's got any other questions, I just thank you for your time. If anybody thinks of anything and goes, "Gosh, I should have asked that," would you be happy for me to just drop you an email with it?
Yes, absolutely. It's been such a pleasure. It's been an absolute pleasure. I hope I've managed to explain the project clearly, because yeah, there haven't been too many questions. So either I've been completely incoherent or maybe quite clear. I don't know which, but
I'm going to go for the latter. I think you've been very clear. It's been very
thank you. But I'd be more than happy to really answer any questions that anyone might have offline. Absolutely.
Brilliant. Perfect. So Jerry, so do you want to tell us a little bit about where the Macular Society research is and bits and pieces?
Absolutely, thank you very much. Yes, the Macular Society has a growing research programme. We've got big ambitions to fund a lot more of this type of research in the future. So this is one of about 20 projects that we have on the go at the moment, all very important, several covering inherited retinal dystrophies and the remainder on age-related macular degeneration. I just want to say that we've got what we call a patient registry, or what we call our research participant database. And it's currently got about 3,000 people on it, and it's where people can register their interest in taking part in research. So Jacqui mentioned that cohort of that selection of patients Doyne Honeycomb Dystrophy that they have at Moorfields. But that won't be every Moorfield, that won't be everybody with the condition around the country. And we have researchers coming to us all the time wanting to carrying out clinical trials, and they need to find patients. And finding rare disease patients is one of the hardest things. So if you think you might want to take part in clinical research, which hopefully we will reach with that stage with Doyne Honeycomb Dystrophy before too long, do go on our website, look in the research section, and sign up. And it's very simple. We don't ask a great deal of information, and then you'll be on our records as having the condition. And if we need to find patients with it in the future, then you will be there and we can come and contact you. We don't provide your information directly to anybody else. We would just match you, tell you about new clinical research that you might be eligible for. So do sign up to the research database if that's what you'd be interested in doing. Thank you very much.
Brilliant. Okay, we're a little ahead of schedule today, which is lovely. So it just gives me the opportunity really to talk a little bit more about the working age service, which we've set up. So the Macular Society has always been supported people with macular dystrophies who are just maybe still be working or a little younger, as I said earlier, with support groups dotted around the country. But a couple of years ago, we decided to have a really good focus on people of a working age, young people and parents of kids with macular dystrophies, to really sort of, because we understand that the needs and the support needed by those groups of people is slightly different. So over the last couple of years, I've been putting together some material and some information around things like, so working age people, things like access to work and in-work benefits, things like PIP and ESA. So we're able to support people with those sort of questions that they might have. This year, we're starting to really concentrate a bit more on the parent support area. So trying to sort of make materials that the parents need to understand central vision loss a bit more in their children. So educational support for teachers and that sort of stuff. So it's a really exciting time. The service is built beautifully over the last couple of years, and we really wanted to push on. And one of the things that we have done to make everybody sort of offer a bit more peer support across the UK for people of a working age and who are a bit younger is I've set up a working age a Facebook group from the Macular Society. It's a closed group, a private group, so you have to request to join. But there's nearly 600 people in there now with all various different kinds of conditions. And I sort of pop lots of information on there from the society's point of view, but also other people are catching up with each other with similar conditions. So if you're feeling like you haven't met anyone with the same condition as you, pop a question on there and you'll be surprised how many people will sort of answer your question or offer support. Which is great. It's a really good resource. It's a really great community, and it's growing every day. And I mean that every day. There's always a new member every day. It's fantastic. Well, so that kind of draws us fairly well to the end. It only leaves me to thank Jerry as usual for your attendance. And Jacqui, really thank you. That was a really very interesting talk. I was very, I find all these talks very interesting. I think where research is going is absolutely amazing. As I say, it does tick my geeky buttons quite a lot. So thanks ever so much for coming. And thank you also for agreeing to answer any email questions that people might think of later. So so that's it really. So thank you all for coming. Next month's virtual clinic is quite a bit exciting as well. Actually, it's looking at Maria Musiji from Moorfields Hospital, looking at Charles-Bonnet syndrome in children, not in adults. So if you've been watching Coronation Street about the Charles-Bonnet story there, oh God, sorry, I can't believe I said that. But anyway, it's about in children. So that's quite an interesting an event as well. So the last Thursday of each month. So that only leaves me to say thank you and good night. Thanks very much.